Welcome back to Health Tech Explorers. Today, we're diving into one of the most quoted but least understood statistical tools in healthcare research, confidence intervals, often written as CI, usually 95% CI. If you've ever opened a medical journal, you've definitely seen them. After means, after risk ratios, after survival estimates. They're everywhere. But what do they really mean? And why are they so important for medicine, pharmacology, and AI-driven health research? Let's break it down. Why confidence intervals? In clinical research, we almost never study the entire patient population. Instead, we take a sample, maybe a few hundred patients in a drug trial, or a few thousand in an epidemiological study. We calculate a statistic, like a mean or percentage, and then try to use that as a barometer for the whole population. But here's the challenge. Our sample statistic is just an estimate, not the whole truth. We need a way to express how close we think this estimate is to the real population value. That's exactly where confidence intervals come in. In essence, a confidence interval puts upper and lower bounds around our sample estimate, saying, we believe the true population value lies somewhere between these limits. For example, let's look at a real-world study. Din and colleagues in 2015 examined maternal HIV seroconversion during pregnancy in South Africa. Out of nearly 9,800 mother-child pairs, they found a seroconversion rate of 3.3%, but they didn't just stop there. They reported a 95% confidence interval of 2.8% to 3.8%. So what does this mean? It does not mean that there is a 95% probability the true rate is between 2.8% and 3.8%. Instead, it means that if we repeated this study 100 times, drawing new random samples each time, about 95 of those studies would produce intervals that truly capture the real population rate. That subtle distinction is hugely important. The true value either is or is not within the interval. But over repeated studies, 95% of the intervals would capture it. Why not just report the mean? You might ask. Why not just say the mean was 3.3%? Because a single number can be misleading. Two drug trials can both report a mean survival of 12 months in cancer patients, but if one trial has a 95% CI of 11 to 13 months and another has a 95% CI of 8 to 16 months, they tell completely different stories about certainty and reliability. A narrow confidence interval suggests precision, often from larger, well-controlled samples. A wide confidence interval warns us of uncertainty, maybe because the sample size was small or the data had lots of variability. For clinicians deciding whether to adopt a therapy, this difference is critical. How confidence intervals are built. Mathematically, CIs rely on the central limit theorem and measures of variability like the standard error. Start with your sample mean, for example the average blood pressure reduction from a new drug. Then calculate how much samples tend to differ from one another. That's the standard error. Around your sample mean, you construct limits, usually so that 95% of the bell curve area is covered. If you know the population variance, you'd use a z-distribution. More often in medicine, we rely on the t-distribution, which adjusts for finite samples. Bottom line, the interval shows the range of values consistent with the data under the chosen level of confidence, typically 95%. Practical implications in medicine and pharma. Confidence intervals are more than just statistical decoration. They directly impact clinical and regulatory decisions, drug efficacy. Suppose a new cholesterol drug lowers LDL by 15 milligrams per deciliter. 95% CI, from 12 to 18. Clinicians can be fairly confident the effect is true and meaningful. Vaccine trials. If a vaccine shows an efficacy of 75%, 95% CI, from 45 to 90, that wide CI tells us the evidence is less precise. Maybe we need a larger trial. Risk estimates, an OR of 2.0 for side effects with a CI of 1.1 to 3.8, suggests a significantly increased risk. But if the CI were 0.9 to 3.8, the true effect might be negligible. Without CIs, we'd risk overinterpreting single estimates without appreciating the surrounding uncertainty. Confidence levels. Now, Why 95%? Why not 99 or 90%? It's a balance. A 99% CI is wider, giving more assurance but less precision. A 90% CI is narrower, offering more precision but a greater risk of missing the true value. The conventional scientific standard is 95%, but regulatory agencies sometimes require other levels depending on context. So, the next time you see a result like mean blood pressure reduction, 10 millimeters of mercury, 95% CI, from 8 to 12, you'll know exactly what it implies. It's not a guaranteed window, it's not a probability attached to a single study, but it is a measure of how reproducible and reliable those results are likely to be. Confidence intervals are the bridge between sample statistics and real-world populations, and in medicine, that bridge determines how confidently we can treat patients, approve drugs, and design interventions. If you found this breakdown helpful, give this video a like, subscribe to Health Tech Explorers, and hit the bell so you don't miss our next deep dive, where we'll go even further into inferential statistics and their role in modern medical AI and clinical trials.